Welcome back to the deep dive. Today, we're getting stuck into something pretty fundamental in modern drug discovery.
We are. It's about how we make sense of these incredibly detailed pictures of cells.
Exactly. We're talking about image-based screening, and specifically this technique called cell painting. It's a cornerstone, right?
It absolutely is. Cell painting, well, it's become the go-to for high-throughput screening. You take your cells, you treat them with something, maybe a potential drug, maybe you tweak their genes.
Okay.
Then you add usually about 5 different fluorescent dyes. These dyes light up specific parts of the cell, the nucleus, mitochondria, the ER, the cell's internal machinery.
So you get this multicolored snapshot.
Precisely. A multi-channel image that captures the cell's physical reaction, its phenotype. It's like taking a unique cellular fingerprint showing exactly how the cell responded to that treatment.
Right, a fingerprint. But, and this is the big but, isn't it? Getting that fingerprint is only step one. The real bottleneck, the challenge you face, is turning that huge, complex visual data into something concise, something quantitative, a number basically, that you can actually use to compare drugs or find matches. And right now that process is, well, it's slow and maybe a bit clunky.
That sums it up perfectly. Current methods, even the really sophisticated ones like, say, DeepProfiler, they involve this whole series of steps.
Okay, what kind of steps are we talking about?
Well, first, you've got to normalize the images, adjust brightness, contrast, that sort of thing. Then comes segmentation. This is a big one.
That's like cutting up the photo.
Exactly like that. You're digitally isolating every single cell in the image. Hundreds, maybe thousands of tiny individual cell portraits. It's computationally intensive.
And I imagine prone to errors if you cut wrong.
It can be, yes. And then you extract features from each tiny cell portrait shape, texture, intensity measurements. And finally, you have to integrate all those features back together to get a profile for the original image or treatment.
Wow, okay, that sounds long and complicated.
It is. It's slow, it adds cost, and each step is a potential place for errors to creep in, which can really limit how reliable the final data is.
So that brings us to our mission for this deep dive. We're going to unpack a newer approach, an innovation called PhenoProfiler, designed to basically tear up that old playbook.
That's the idea. PhenoProfiler is what we call an end-to-end model.
Meaning?
Meaning it skips a lot of those intermediate steps. You feed it the whole multi-channel image, the entire picture, no cutting up, and it directly outputs that low-dimensional feature vector, the quantitative profile we need, all in one go.
Okay, that sounds much more streamlined, but does it work as well? What's the promise here?
The promise is twofold. Efficiency, definitely, but also critically better performance. The research suggests PhenoProfiler can be significantly better, perhaps up to 20% better in some key metrics at identifying compounds that are biologically related compared to the older methods.
And when we say better performance in this context, we need ways to measure that biological relevance, right?
Absolutely. We rely on specific metrics. 2 key ones you'll hear about are folds of enrichment, often just called FOE.
FOE, got it.
Which measures how well the model groups together treatments that have the same biological mechanism, the same way of working.
So does it put all the drugs that target the same protein pathway together?
Exactly. A high FOE means it's successfully finding those connections. The other main one is mean average precision, or MAP. MAP gives you a broader view of the ranking quality. It balances finding relevant things, precision, with finding all the relevant things, recall. But FOE is often the hero metric here.
Because a high FOE tells you the model isn't just seeing random patterns in the images.
Right.
It's actually understanding the underlying biology and grouping things that, biologically should be grouped together.
That's the goal. You want the features it extracts to reflect true biological similarity.
Okay, that sets the stage perfectly. Yeah. Let's get into the nuts and bolts of how PhenoProfiler achieves this. The big differentiator, as you said, is this end-to-end approach. It skips segmentation, that whole cutting up the photo business.
It does. It takes the entire image, warts and all, as its input. And to show this wasn't just a theoretical idea, they tested it on a massive scale.
Oh, massive.
We're talking over 230,000 whole slide images used just for the end-to-end validation, plus millions more single-cell images for other comparisons. They use large, publicly available datasets like BBBC022, CDRP Bio, TARF, really put it through its paces.
Okay, huge scale, but here's what I'm wondering. If you skip the step where you carefully isolate each cell, how does the model properly see the important details, like the cell edges, the morphologies? Aren't traditional methods kind of optimized for looking at those isolated objects?
That's a great question, and that's where the first piece of PhenoProfiler's specific architecture comes in, the gradient encoder.
Gradient encoder, okay, what's its job?
Its specific job is to really enhance the information about cell edges and boundaries within that whole image context. To do this, they use a clever twist on standard convolution, something called difference convolution, or DC.
Difference convolution, how is that different from, well, normal convolution?
Okay, so in standard convolution, you slide a filter over the image, and for each position, you calculate a weighted sum of the pixel values in the filter's neighborhood. Pretty standard deep learning stuff.
Right.
Difference convolution adds a crucial tweak. Instead of just summing the neighbors, it explicitly calculates the difference between each neighboring pixel and the center pixel under the filter. Then it calculates the weighted sum of these differences.
Ah, so it's inherently looking for changes, for contrast.
Precisely. The formula looks something like DOC, it's some WOX. That theta value controls the balance. If theta was 0, you'd just get traditional convolution back. But by setting theta to a positive value, you force the calculation to emphasize gradients.
So it mathematically sharpens the image wherever there's a boundary, like the edge of a nucleus or the cell membrane.
That's exactly the effect. It makes the morphological details, the shapes and structures that often change with drug treatments, much clearer for the subsequent layers of the network.
Clever. Is that all the gradient encoder does?
There's a bit more structure. It actually uses two parallel branches of this difference convolution. One set up to be sensitive to stronger gradients, sort of deep changes, beta 1 to sorbing mothers 7-7, and one for shallower ones.
Okay, so it captures contrast at different scales.
Kind of, yeah. The outputs from these two branches are then combined, passed through a standard MLP layer, and then fed into a ResNet 50 block.
ResNet 50, that's a standard workhorse for image analysis.
It is. Using a pre-trained ResNet helps extract robust hierarchical features, and importantly, the residual connections in ResNet help prevent the vanishing gradient problem, ensuring the network can learn effectively even though it's deep.
So quick recap. The gradient encoder uses difference convolution to specifically highlight cell edges and morphology within the whole image, then uses ResNet to pull out rich features from that enhanced view.
You got it. It focuses on extracting high-quality local and hierarchical information first.
Okay, but cells don't exist in isolation, even within the image. How does PhenoProfiler understand the bigger picture? The relationships across the entire field of cells. Maybe how changes in one area relate to changes somewhere else.
Right, good point. You need to capture that context. That's where the second major component comes in. The transformer encoder.
Transformers.
Yeah.
They're everywhere now, from language models to vision.
Indeed, and they're particularly good at capturing long-range dependencies. After the gradient encoder has done its job extracting those detailed local features, the transformer steps in to look at the global picture.
How does it do that in an image context?
It uses what's called a multi-head self-attention mechanism. You can sort of imagine it like this. For any given part of the feature map representing the image, the attention mechanism allows the model to look at all other parts simultaneously.
Okay.
And it learns to calculate attention scores, basically, how relevant each other part is to understanding the current part. The multi-head part just means it does this process multiple times in parallel, looking for different kinds of relationships.
So it's like giving the model peripheral vision across the whole image. It can see how, say, something happening with mitochondria over here might relate to the nucleus shape way over there.
That's a great analogy. It figures out which relationships matter no matter how far apart they are in the image. This ensures you capture not just individual cell features, but also the contextual information of the entire cell population in the well.
And does it have things like layer normalization and residual connections too?
Yes, those are standard components within the transformer blocks as well. They help stabilize the training and ensure the gradients flow properly, just like in the ResNet. It refines these high-level features that represent the whole image.
Okay, so we have difference convolution, sharpening the local details via the gradient encoder, and then the transformer, understanding the global context and long-range relationships. That sounds like a really comprehensive way to see the image.
It's designed to be, yeah, extracting the most informative features possible from the raw pixels.
But just extracting features isn't enough. The model has to learn what those features mean biologically, right? How does it connect those features to actual drug effects? This takes us to the learning strategy, doesn't it? The multi-objective learning module.
Exactly. And this is another area where PhenoProfiler takes a really interesting and quite different approach.
Different how? What's the usual way?
Typically, in these kinds of models, you train them primarily as a classification problem. You have labels. This image is from cells treated with drug A, this one with drug B, this one is a control. The model learns to predict the correct label.
Okay, seems logical. What's the downside?
The downside is that biological reality is often much more nuanced than discrete labels like drug A or drug B. Relying only on classification can limit the model's ability to pick up on subtle but important morphological changes.
So if drug C causes a change that's maybe halfway between the effective drug A and the control, a simple classifier might struggle, or just lump it in with one or the other.
Precisely. It makes the model less sensitive to fine-grained differences and potentially less generalizable when you take it to a new experiment, maybe with slightly different conditions or concentrations. Classification can be too blunt an instrument.
So PhenoProfiler does something else, or something more.
It does something more. It uses a multi-objective approach, training the model to satisfy 3 distinct learning goals simultaneously. The idea is to create a richer, more biologically meaningful feature space.
3 objectives. Okay, what are they?
Well, first, it does include the standard classification learning. So it still learns to predict the treatment condition based on the ground truth labels using standard cross-entropy loss. That provides a basic anchor.
Okay, so it doesn't throw the baby out with the bathwater.
Not at all. The third objective is also relatively common in modern representation learning. Contrastive learning.
Right, contrastive learning. That's about pulling similar things together and pushing different things apart in the feature space.
Exactly. It encourages the learned features embeddings for treatments that are biologically similar, example, same mechanism of action, to be close together, while pushing embeddings from dissimilar treatments further apart. This helps build robustness and generalization, especially if some labels are noisy. It focuses on relative relationships.
Makes sense. Classification for predicted categories, contrastive for robust grouping. What's the crucial second objective then? This sounds like where the real innovation lies.
It really is. The second objective is regression learning, Math-Cal MSE, and this is designed specifically to capture those subtle morphological changes we talked about.
Regression, so predicting a continuous value, not just a category.
Yes. Instead of just relying on the discrete treatment labels, they leverage existing, rich, continuous feature information as a supervisory signal.
What kind of continuous information? Where does it come from?
They used pre-computed median morphology profiles from a large public dataset specifically, one called CPG0019, which contains cell painting data processed with CellProfiler. These profiles are essentially detailed quantitative descriptions of average cell shape, size, texture, etc. for many different treatments.
Oh, so they're using existing, highly detailed quantitative measurements of cell appearance as a kind of expert guide for the model.
That's a good way to put it. The model is trained using mean squared error, MSE loss, to predict these continuous morphological profile values based on the image it sees.
So it's not just learning this looks like drug A, it's learning this looks like a cell with this specific quantitative profile, a profile that reflects maybe a certain amount of nucleus rounding or mitochondrial intensity.
Precisely. It forces the model to learn the degree of change, the subtle shifts in morphology, far more accurately than just classifying into broad categories.
Okay, that sounds powerful.
Yeah.
But now you have 3 different goals potentially pulling the model in different directions: classification, contrastive grouping, and this fine-grained regression.
Yeah.
How do you balance them? It sounds like a potential tug of war.
It could be, yeah. The key is how you combine them in the final loss function. It's a weighted sum. Mathical total, lambda1 mathical CLS, plus lambda2 mathls, plus lambda3 mathical. Those lambda1, lambda3, 3 are the weights assigned to each objective.
And finding the right weights is crucial, I assume. Did they figure out the optimal balance?
They did, through sensitivity analysis, basically, trying different combinations and seeing what worked best. And the results were, well, quite striking.
How so?
They found the optimal weight for classification was a lambda1 missense. For contrastive learning, lambda-pre, it was 1. But the weight for the regression loss, lambda2, was 100.
Hold on. 100. As in 100 times more important than the contrastive loss and 1,000 times more than the classification loss.
That's what the optimization pointed to. A massive emphasis on the regression component.
Wow. That's That's not subtle. It's like the model is being told. Forget mostly about the labels. Forget mostly about just general grouping. What really matters is getting these quantitative morphological profiles right.
It strongly suggests that accurately capturing the degree of morphological change is paramount for building a useful representation in this context. And the ablation studies back this up dramatically.
Ablation studies where they remove parts of the model to see what breaks.
Exactly. When they removed the regression learning component entirely, leaving only classification and contrastive learning, the performance plummeted. That key PhenoE metric, the measure of biological grouping, dropped by a massive 16.3%. Average precision also fell nearly 6%.
A 16% drop in PhenoE just from removing the regression. That's huge. It really proves that learning the quantitative degree of change is vital. But why? Why is it so critical, especially for discovering, say, new drug mechanisms?
Well, think about it. Drugs with truly novel mechanisms, or maybe drugs that have very subtle effects, they're unlikely to produce a phenotype that screams, I belong in category X. They won't fit neatly into the predefined classification buckets derived from known drugs.
Right, they might create a new kind of fingerprint or just a slight smudge on an existing one.
Exactly. They often present as subtle shifts along a continuum of morphological changes. By forcing the model to learn to predict these continuous values via regression, PhenoProfiler essentially learns a much more sensitive and nuanced feature space. It becomes better at distinguishing those small but potentially critical biological differences.
So it's better equipped to spot something unexpected, something that doesn't fit the old patterns, because it's learned the language of subtle quantitative change.
That's the core idea. It makes the model inherently more powerful for discovery, not just for confirming known effects.
That's fascinating. So just to quickly summarize this learning part, PhenoProfiler uses 3 objectives: classification, contrastive, and regression, but puts enormous weight on the regression part, forcing it to learn the degree of cell change based on quantitative profiles. And this seems key for its ability to group biologically related compounds effectively.
Spot on. It builds a feature space that's highly sensitive to the magnitude of the biological response.
Now, if you, our listener, were thinking about which objective is most crucial for spotting a brand new drug mechanism, something maybe never seen before, based on this evidence that heavy weighting strongly suggests learning the magnitude of the cell's response via regression is probably the most important piece.
It seems to be the secret sauce for capturing nuance. And this ability to capture nuance ties directly into the results, particularly how PhenoProfiler handles another major headache in this field.
Right. Let's talk results and robustness. We mentioned the performance gains earlier, like that huge 64.9% relative jump in PhenoE over dProfiler on one dataset. Impressive numbers. But high performance doesn't mean much if the data is messy or inconsistent, does it?
Not at all.
Yeah.
And in high-throughput screening, probably the single biggest source of messiness is the batch effect.
Ah, yes, batch effects. The bane of many large-scale experiments. Can you remind us what that is?
Sure, batch effects are systematic variations introduced by the experimental process itself, rather than the biology you're trying to measure. Things like slight differences in reagent batches, incubation times, maybe even which microscope or plate reader was used.
So cells on plate A might just look slightly different from cells on plate B, even if they got the exact same treatment, just because the plates were processed slightly differently.
Exactly. And these technical variations can be large enough to completely obscure the real biological differences you're looking for. Your data can end up clustering by plate, not by treatment effect. It's a huge problem for reproducibility and reliability.
How does PhenoProfiler deal with this? Do they have a complicated correction step?
Actually, this is another elegant part of the system. They have an optional but highly effective optimization step that's applied during inference, meaning after the model is trained, called the phenotype correction strategy, or PCs. And it's virtually cost-free to implement.
PCs. Okay. Applied after training.
Yeah.
How does it work? Is it complex?
The concept is surprisingly simple. Once the model has generated the feature embedding for a particular treated well, a particular plate, PCs refines that embedding. It does this by subtracting a weighted average embedding of all the control wells from that very same plate.
Wait, so for each treated well, it subtracts the average look of the untreated cells from the same plate?
Essentially, yes. The formula is OPI O for we 10. By subtracting the plate-specific control baseline, you're emphasizing the relative change induced by the treatment within that plate's specific context.
Ah, I see. So instead of representing the absolute morphology of the treated cells, which might be affected by the batch effect,
Right.
it represents how different those cells are from their own baseline controls on that specific plate. It cancels out the shared plate effect.
That's precisely the intuition. It focuses the feature representation on the treatment-specific deviation from the norm for that batch, making it much less sensitive to the overall plate-to-plate variations.
That makes a lot of sense. It's anchoring the measurement locally. Does it actually improve the biological grouping, the FOE?
Consistently, yes. Applying PCs generally leads to better FOE scores, indicating that this relative representation is more biologically meaningful.
Okay, so PCs is a smart post-processing trick. But does the core PhenoProfiler model itself do a better job with batch effects, even before PCs? You mentioned that regression loss seemed important.
That's the really interesting finding. It seems PhenoProfiler learns representations that are inherently more robust to batch effects, likely thanks to that multi-objective learning, especially the heavily weighted regression component.
How can we measure that inherent robustness?
There's a metric for that too, called the inverse median absolute deviation, or IMAD. You calculate the embeddings for the same type of sample, for example, all the DMSO controls, across many different plates. Then you measure how spread out those embeddings are in the feature space.
Okay, so low spread means the controls look similar regardless of the plate.
Exactly. A tighter cluster means less batch effect. IMAD is the inverse of that spread, so a higher IMAD score means better batch effect correction, but the representations are more harmonized across plates.
Right. So how did PhenoProfiler stack up on IMAD compared to, say, Deep Profiler?
The difference was quite stark. On the BBBC022 dataset, Deep Profiler's raw embeddings had an IMAD of only 0.326. Even after applying a separate, dedicated batch correction algorithm called typical variation normalization, Deep Profiler only reached 0.458.
Okay, so even with extra correction, it was still somewhat spread out.
Right.
What about PhenoProfiler?
PhenoProfiler, without any extra correction beyond its own training process, achieved a much higher IMAD of 0.603.
Wow, so significantly tighter clustering straight out of the box.
Yes. It suggests that the way PhenoProfiler learns, particularly that focus on quantitative regression against stable morphological profiles, allows it to intrinsically learn features that are less susceptible to technical variations between plates. It's building in the robustness during training.
That's a massive advantage. It means potentially less need for complex and sometimes problematic downstream correction steps. The pipeline becomes simpler and more reliable.
Precisely. It learns harmonized representations inherently.
And did they visualize this? Can you actually see the improved clustering?
Yes. They used UMP projections, a common way to visualize high-dimensional data in 2D. And the visualizations confirmed the metrics. PhenoProfiler's embeddings showed clear clustering of treatments based on known biological relationships.
Like what?
For compound treatments, like in BBC022, treatments with the same known mechanism of action, MOA, clustered tightly together. For datasets involving genetic perturbations like TORF BBC037, which looks at gene overexpression, treatments affecting the same signaling pathways like MAPPK or PI3, KKKT, formed distinct clusters.
So the pictures confirm the numbers. The features PhenoProfiler extracts aren't just abstract vectors. They genuinely capture in group treatments based on shared biology. It's effectively translating pixels into biological insight.
That's the takeaway. It demonstrates the biological relevance of the learned representations.
Okay, this has been a really deep dive into PhenoProfiler. Let's try and pull the main threads together.
Sounds good. I think the major contribution here is a shift towards a truly scalable and robust pipeline for phenotypic profiling. It moves away from that complex, error-prone, multi-step process involving explicit segmentation.
Right, the cutting up the photo part.
To a single, end-to-end model. Key to its success are, 1, the architecture using difference convolution and transformers to effectively process whole images, and 2, the multi-objective learning strategy.
And within that learning strategy, the real kicker seems to be the huge emphasis on regression learning, teaching the model the degree of morphological change using continuous quantitative profiles.
Absolutely. That heavy weighting on the MSE loss seems critical for capturing subtle biological nuances and, importantly, for learning representations that are inherently more robust to batch effects, as shown by the AMAD scores. The PC strategy then provides an extra simple refinement step.
So overall, it delivers better biological grouping, higher FOE, improved robustness, higher IMAD, and does it more efficiently by being end-to-end. It transforms these complex images into low-dimensional representations packed with biological meaning.
That's a great summary.
And importantly for you, the listener, this isn't just some theoretical concept locked away in a lab. The creators have made it accessible.
That's right. PhenoProfiler is available as a Python package for those comfortable with coding. But they've also set up a user-friendly web server, phenoprofiler.org.
Meaning, even if you don't have deep coding expertise, you could potentially upload your own cell-painting image data and leverage this advanced analysis.
That seems to be the goal, yes, to make state-of-the-art phenotypic profiling much more broadly accessible.
Fantastic. Now, looking ahead, PhenoProfiler clearly represents a big step forward in analyzing cell morphology from images. But where does this field go next?
Well, while PhenoProfiler excels at extracting information from the image, the ultimate goal in drug discovery is often deeper mechanistic understanding. The discussion around the paper suggests future work will likely focus on integrating these powerful morphological profiles with other types of biological data.
Like combining what the cell looks like from PhenoProfiler with, say, what genes are turned on or off.
Exactly, or maybe integrating it with information about the chemical structure of the drugs themselves. Combining these different data modalities holds the promise of unlocking much richer insights into why a drug causes a particular phenotype, not just identifying the phenotype itself.
Which leads us perfectly to our final provocative thought for you to mull over. We've seen today how PhenoProfiler, particularly through its heavily weighted regression loss, excels at quantifying the precise degree of morphological change in cells. The next step is integrating other data to explain the why behind that change. So if you were designing the next generation of this kind of analysis, aiming for the absolute deepest mechanistic insights, which type of non-image data would you prioritize integrating first? Would you combine the morphological profiles with the detailed genetic expression profiles resulting from the treatment? Or would you focus first on integrating the raw chemical structures of the compounds being tested? Which combination do you think holds the most promise for truly understanding how a drug works at a fundamental level, based on the changes PhenoProfiler quantifies?
A fascinating question. Genetics or chemistry? Where do you place your bet for the next big leap in understanding? Something to think about.
Definitely food for thought. Thank you for joining us for this deep dive into PhenoProfiler. We'll see you next time.